Hi, everybody, and welcome back to our final part when it comes to our genetic and inheritance lecture. So, what we're going to be looking at today is going to be the non-Mendelian genetics. So, as of right now, we've learned a lot of different rules when it comes to genetics, exactly how to predict the offspring, see exactly how certain traits are passed on from one generation to the next, and exactly what those rules are when it comes to those genetics, when it comes to dominant and recessive traits. Now, there's always exceptions to the rules. And in this case, what we're going to be looking at is something known as non-Mendelian. So in this case, they although follow the rules, they still end up with a specific or different type of outcome. And we're going to kind of see what those differences are and how to identify them and how to utilize them when we're doing our Punnett squares and when we're figuring out what our ratios are going to be. So one of the very first ones we're going to look at is something known as incomplete dominance. So when it comes to incomplete dominance, just like the name suggests, the dominant allele is not necessarily going to be shown. Now, I know that sounds kind of confusing because I'm sure you're like, well, Mr. Chang, I thought you said that the big letter is always dominant and they always show over the recessive. And again, that is usually true when it comes to our Mendelian. But in incomplete dominance, whenever we have a dominant gene paired with a recessive gene, neither of them are going to show. We're going to have something more of a blend between the two. So let's see exactly what I'm talking about. Looking at this over here, this is going to be a great example when it comes to incomplete dominance. This is going to be known as a snapdragon. Now, snapdragons can come in a variety of different types of colors. Now, if we're crossing a true breeding red flower right here, that's going to be FF. And right off the bat, you can see F is a big letter, so we know that that's going to be a dominant gene. And we can also get true breeding white flowers right here, and you can see they're little f's. So that means that's going to be a recessive gene. But whenever we go ahead and mix these together, we end up with a different phenotype altogether. In this case, we have the color pink. So anytime we have heterozygous, in this case, big F, little f, we're ending up with a blend of the two. So again, if we wanted red, we would need big F in order to get red. If we wanted white, we would need little f, and that's how we would get white. And whenever we have heterozygous, so we have one big F and little f, we would get pink. And this is why this is known as our incomplete dominance. Because again, we know we have a dominant red here, we know we have a recessive white here, but whenever we end up with heterozygous, we end up with something that's completely different. Again, incomplete or neither of them showing. In this case, we have something more of a blend between the two, giving us the color pink. So looking at this, you can see exactly how this is going to kind of show and what exactly is happening. So again, if we go back to our example and we're looking at our true breeding red flower, so big F right here for our P1, and then we have our true breeding white flower, little f for our P2, you can see that we're ending up with a 100% all heterozygous, and they're all going to be pink. Again, the reason why they're pink is because they contain one dominant gene and one recessive gene. And again, because this is an incomplete dominant problem, you can see that neither phenotype is showing. It's going to be something different, in this case, the color pink. Now, if we were to take two heterozygous right here with our P1, P2, you can see we're ending up with a 1 to 2 to 1 ratio. Right here, big F, giving us the color red, or in this case, a 25% chance. You can see we have heterozygous here, big F, little f, giving us a 50% chance right here for the color pink. And then finally over here for white, we would have little f, giving us a 25% chance. So to kind of summarize exactly what incomplete is you'll have a dominant like red over here, and you'll have a recessive like white over here. But whenever you have a heterozygous, you end up with something with a mix between, something like the color pink, which is going to be heterozygous big F, little f. So that's kind of the gist when it comes to our incomplete dominant problems. All right, so the next one we're going to look at is going to be something known as multiple alleles for a single trait. So in this case, the example we're going to be looking at is blood type, specifically ABO blood types. Now, as you know, there are many different blood types that people can have. You can have blood type A, just like this here, blood type B, just like this here, AB right here, and then type O. Now, one of the big important things when it comes to this is exactly the antigens that are going to be found on top or on the red blood cells. These are going to be kind of flags for those that you can be found on the surface of the red blood cells. Now, the reason why this is so important is because it's really important to understand exactly what blood people can take in and what blood people can actually accept. So if we were to take our allele A, it would cause problems, or it would cause production of a antigen A on the red blood cells. B allele will cause production of B antigen on red blood cells. And if we have both A and B allele, then we're getting both of those A and B right here. And then if we have type O, then we don't have any antigens at all. So again, this is going to help dictate exactly who can receive what type of blood. In this case, we would know that the type AB would be the universal recipient because they have both A and B alleles for the antigens. And we can see that type O blood, since we have the absence of it, would be known as our universal donor because again, it can give to everybody because it has the absences of these antigens. So now let's go ahead and see exactly how we can get these different types of blood and exactly what this means and how it relates to our multiple alleles. So the reason why this is known as multiple alleles is because we can end up with many different types of blood, and there are very specific genotypes we need in order to get those types of blood. So in this case, you can see we have our blood types A, B, and O here, A, B, and O, and you can see that we have many different types. So the important thing to kind of know is that A and B are both going to be dominant and O is going to be recessive. So that means if I wanted blood type A here, then I would have to have A and A or AO. This would give us blood type A. So we can have blood type AA, homozygous dominant, or we can have heterozygous AO. So you can see we have two ways of getting that. If you wanted blood type B, we would need BB, which you can see right here, or BO, which would give us blood type B. In order to get blood type O, we know that it's going to be recessive, so that means the only way to get this would be OO. Now, there's one more blood type that we can end up having, and this would be our blood type AB. Now, this is where we see codominance happen. So unlike incomplete, where neither of them are showing, when it comes to codominant, just like co-ed, you can have both show. So in this case, if we have A and B come together, we would end up with blood type AB, which you can see right here. So again, they're both showing. So because blood type or the alleles A and B are both dominant over blood type O, these would be the ways to get that. To get A, it'd be AA, homozygous dominant, AO, heterozygous, or BB, homozygous dominant, or BO, heterozygous. In order to get O, you would need both recessives, OO, and then in order to get blood type AB, you would need both A and B, and since they're both dominant, both of them are going to show because they are going to be both codominant. So in this case, the genotype would be A and B. All right, so let's go ahead and test our knowledge with the new Mendelian, non-Mendelian genetics we just learned. So just as always, go ahead and take a second, pause this, and then we'll go ahead and check this out together. All right, welcome back, and let's go ahead and see how everybody did. So in number one, it says, what are the possible genotypes for an individual with blood type A? Select all that are appropriate. So right here, we know that we're dealing with a multiple alleles problem. So when it comes to blood type, we can have blood type A, B, and O. In order to get A, it would be AO. To get blood type B, we know it's B, O. In order to get blood type AB, remember this is going to be codominant, so it's going to be AB. In order to get blood type O, it's OO. So looking at this, we know it's going to be blood type A, and we're just identifying the different genotypes we can have in order to get blood type A. So right here, we would know we would have a homozygous dominant, so we would know it would be AA, and we know we would have heterozygous AO. It could not be AB because if it was, then it would be blood type AB. And since O is recessive, it's going to be dominated over with the A. So in this case, it would be AO. What are the possible genotypes for an individual with blood type AB? So we're going to go back again and check this out here. we know that in order to get blood type AB, we would need both dominant genes A and B. And since it's codominant, both of them are going to show. So that means it's going to be this one right here, AB. A woman with blood type O, so we know O right here, the genotype is going to be OO, has a child with a man with blood type B. In this case, we know it's either going to be BB or BO. What are the genotypes of the parents? Use the Punnett squares to help you determine the possible genotypes and blood types / phenotypes of the child. So again, we know the mom is blood type O, so it's going to be OO, and the dad can either be BB or BO. The reason why it can either be both of these is because we're not quite sure. We know we have blood type B, but there's two ways of getting it, which are going to be right here. So this is why we need to use two Punnett squares. So we're going to go ahead and do this. This will be our P1. This will be our P2. So P1 goes up here. P2 goes off to the side. And then just like previously, we're just going to do a regular Punnett square. So these are going to come down just like that. These are going to go over just like that. And then we're going to end up with this right here, which is going to be 100% B. So we have blood type B. And then we know it's going to also be 100% BO. For this one, we have B or OO and then BO. So unlike this, where it was homozygous dominant, we're looking at heterozygous now. Do the exact same thing, cross those out or cross them down, and you can see we have a 50% chance of it being blood type B. So B equals 50%. And over here, you can see we have blood type O, which is going to also be a 50%. So the possible genotypes for the child can either be BO or OO, depending on what the father is. And then possible blood types / phenotypes for the child, again, is either going to be blood type B or blood type O. Again, all depending on what the father is, because we're not quite sure what the genotype is for his blood type B. If it was BB, it would be 100% blood type B with the genotype BO. And then if it was BO, so if he was heterozygous, then it would be a 50-50 split. All right, same thing. Go ahead and give this a pause and then give these a try. All right, welcome back. For these, I'm not going to go ahead and go over these. So just go ahead and read the ones in red to see if your answers match with these. All right, so now what we're going to be looking at is pedigrees. So what we're going to do is we're going to see inheritance of autosomal recessive genetic disorders. So as you know, genes aren't just regulated to hair color, eye color, things like that. It's essentially everything within our body. So we're going to see exactly how we can kind of pass these down and exactly where they're coming from. So one of the first things we need to identify here is going to be this. So what this is going to be known as is a pedigree. What a pedigree does is a visual representation of how certain genes or traits are passed on. So right here, you can see we have Jim and Jane. Jim is going to be a square. Squares are going to be males. And then Jane is going to be a circle, which are going to be females. Now, one thing that's important is typically when we're dealing with a pedigree, we'll have a shaded in portion. If we're tracking something very specific. In this case, our shaded portions are going to represent sickle cell, just like this over here. Now, if it's shaded, that means that person has sickle cell anemia. If they're not shaded, then that means they are normal. And then if they are a carrier, then that means they're going to be half shaded in. So what we're essentially going to do here is we're going to go ahead and identify and see how we can do this Punnett square to see what their kids are going to look like. So it says both Jim and Jane are carriers for sickle cell anemia, a recessive genetic disorder. So just like that, we learned something incredibly important. We know that they are going to be recessive. So what we're going to do is we're going to go ahead and pick some letters to represent that. So I'm going to go ahead and do, let's say, a big S, which is going to equal dominant and normal. And then our little s, which is going to be for sickle cell. So I'll put an SC for sickle cell. So in this case, we know Jim over here is a carrier for the disorder, but he is not showing it. So in this case, we're going to go and put a big S, little s here. Because again, we know Jim is a carrier. So although Jim does not have it, he still has the genes to pass it on. Same thing with Jane. Jane is shaded, so that means we know that they are a carrier. So we're going to go and put a big S here and a little s down here. So just like that, based upon this, we know what our genome and phenotypes are. So in this case, we're going to go and plug this into our Punnett squares. So we can have a big S here, little s, and then we can have a big S here and a little s. So what we're going to do now is we're going to see what the probability of the chances of their kids having sickle cell anemia. So we're going to go and bring this down. So we're going to put S here, big S, little s, and then we're going to have a big S, and then little s, little s. Now remember, in order to get normal, you just need one big S, and then it doesn't matter what the second one is. If it's a big S, they're going to be full normal. If it's big and little, they're going to be a carrier. And if it's little, then that means they're going to have sickle cell. So looking at this, you can see that we have one big S. So for our ratio, so let's do pheno and geno right here. And then for pheno, we'll put normal and then sickle cell right here. So looking at this, we have 3 that are going to be normal, and we have 1 that's going to have sickle cell. For our phenotype. For our genotypes, we have SS, big S, little s, and 2 little s's. In this case, it's going to be a 1 to 2 to 1 ratio. So the reason why this is different is because we have that special carrier. So in this case, they have a 75% chance of a normal kid, and then they have a 25% chance of somebody having sickle cell anemia. But when we look at the genotypes, we have a 25% chance of them being 100% normal. We have a 50% chance of them being a carrier. And then finally, we also have a 25% chance of them having sickle cell anemia. So that's what that should look like. So they're going to end up with about a 25% chance if they ended up having kids. So this right here is showing us that they did end up having 2 kids, and that these kids have a 25% chance right here of having sickle cell anemia. All right, so looking at this over here, this is going to be another predicting pattern. So Punnett square of the pedigree below shows the inheritance of sickle cell anemia in the family. So why can sickle cell skip traits during the generations? So one of the big reasons why is because sickle cell is going to be a recessive condition. So because it's recessive, it's not necessarily going to show up very often. Remember, there's a very specific genotype that needs to be met in order for that individual to have sickle cell anemia. So remember, if they have 2 big S's, they're going to be normal. If they have a big S, little s, they're going to be normal, but they're going to be a carrier. Meaning that although they don't exhibit sickle cell, they still have the ability or the genes, because of this one, to pass on to their offspring, and they may end up having sickle cell. And then the only way to get it is you must have 2 recessive genes. It has to be homozygous recessive for that person to have sickle cell. So you can see that there is a much higher probability or a much higher chance, in this case, usually about a 75% chance of an individual being normal and not having sickle cell. So you can see right here, if we were to take this person, cross it with this person, you can see that all of their generations right here, so generation 2 and 3, almost everybody doesn't have it except for this one person. And again, the reason why is because this specific requirement needs to be met. So again, there isn't a high chance for these to kind of show up. So that's why we end up kind of seeing some skipping. We can also see skipping too, depending on what their genotypes are here and here. So if this person was a homozygous dominant for normal, and this person was a homozygous recessive for carrier, then that means everybody here would be a carrier itself, but they would all be normal. And then they would go ahead and have offspring or have kids with somebody else. And again, depending on who these people are right here and here, what their genotypes are is going to dictate the probability of what they're going to have next. So again, if this person was homozygous dominant for normal and this person was a carrier, then again, there would not be a possibility for any of these kids to have it. The only possible way would be this person be a carrier, this person be a carrier, in order for this person to have that homozygous recessive trait in order to have sickle cell. So again, when it comes to those recessive traits, it's not necessarily in the favor of that trait, of that recessive trait to show up often. All right, so we're going to go ahead and look at another example of this. What we're going to be looking at is Huntington's disease. So when it comes to Huntington's disease, this is a genetic disorder that is going to be characterized by degradation and death of neurons in certain areas of the brain. So it can be shown, or some of the symptoms include sudden jerking, involuntary movements throughout the body, severe problems with balance, coordination, difficulty shifting your gaze, moving your head, hesitating, slurred speech, dementia, all of those things. And you can see that this is a normal person right here. And you can see this is somebody with Huntington's right over here. So you can notice that the regions of the neural degradation appears black in the image. And you can see that we have all of these spots right here. Again, those are all death of neurons. And this is causing all of these because now we have difficulty sending those signals. So Huntington's is caused by a mutation in the Huntington's gene. And this leads to the production of a mutated protein that gains an abnormal function and causes that neuron death. So again, remember, you can see that this is that gene right here that's going to produce this protein. And we want this protein to look this specific way. This protein is going to give us our healthy neuron. Now, whenever we have that mutated, you can see we have an elongated portion over here versus this short portion that we have here. And you'll notice that we almost have double the amount here, meaning that it's going to give us that neural degradation. So let's go ahead and take a look at how we can predict to see Huntington's. So in this case, it says Jack just found out that his father has Huntington's disease, a dominant genetic disorder that also afflicted his grandmother. Jack's family's pedigree is shown below. So what is the probability of Jack will develop this disease and his sister? So over here you can see male right here. We see females, circles. Huntington's is what we're tracking. So this is what's shaded in. So we can see this person has that disorder. You can see normal. These people don't. So we're going to go ahead and try to figure this out by doing our Punnett square. So looking at this over here, you can see, remember, it's a dominant trait, a dominant genetic disorder. So in this case, the only way to get it would be big H, big H. We know that Jack's father has had it, right? So we know that he's going to be big H, right here. And we knew that it was also afflicted, disorder that also afflicted his grandmother. So what we're doing here is we're going to go ahead and cross this. So right here is that specific generation we're looking at. Because remember, we're dealing with the male, and we know that it's Jack's father. So we know it's big H, and then we're going to do little h, because the mom does not have it. The reason why we know it is a little h and not big H, is because, remember, it is a dominant genetic disorder. So that means if it's big H, then they're going to have Huntington's disorder. If we had big H, we still have that dominant gene, so that's going to give us Huntington's. So the only way to not have it is two little h's, or in this case, our homozygous recessive. So in this case, we would just go ahead and plug these in. So we have our P1, P2 over here, and then we're going to go ahead and cross this right here. And you can see we have two heterozygous. So heterozygous for big H, little h, or a 50%. And then over here, you can see we have two little h's, giving us another 50%. So in this case, remember, this is a dominant genetic disorder. So that means once you have that big letter, that means that you are going to have that Huntington's disease. So you can see that there's a 50% chance. And then again, little h, there's a 50% chance this person will not. So in this case, when it comes to this, Jack has about a 50% chance of having it, as well as Jill. All right, so let's go ahead and look at another one. The pedigree below shows the inheritance of a Huntington's disease in a family. So why does Huntington's trait appear in every generation? So one of the big reasons why is because it is a dominant trait. So again, because it's dominant, all you need is that one big H. It does not matter what the other one is, and you're going to end up with that Huntington's disorder. The only way you cannot have it or not get it is two recessive. So in this case, little h, So again, if this person was little h, and this person was big H / big H, little h, again, it's going to still give them the opportunity to. So again, if we were to do that Punnett square, you would see that there would be a more or a higher probability of somebody having it. In this case, since we know we have two people that have it, most likely this person had a big H, little h, because again, if we had homozygous dominant and heterozygous and we cross this, a big H would show in every single one, meaning that everybody would have it. So in this case, we have big H, little h. You see it showing up here. They have it. So if they end up having kids, again, they're all going to be carriers, or they could be homozygous dominant for it. But again, you can see it's now here and then here. So again, one of the big reasons why is because it is a dominant condition. So you just need that singular big H. Unlike sickle cell, where it was recessive and was much less likely for it to show up, Huntington's, because it's dominant, it's much more likely to show up. All right, and that is going to conclude our lecture. So as always, if anybody has any questions, comments, concerns, please feel free to reach out. And I look forward to seeing everybody in our next lecture.